So today we are going to start talking about microbiology. Micro being really small, biology, living things. So it's the study of small life. Just before we start, let's go ahead and talk about the microorganisms that are harmful. It's what most people think of when they think of microorganisms. Bacteria, things that invade, cause infection, all that kind of thing. But harmful microorganisms only account for about 1% of all microorganisms. Most microorganisms are beneficial to humans. Now, as we look at the spectrum of light over here on the left, some of the things you might notice are, radio and TV waves are over here. And X-rays that we use for imaging are over here. But there's a small portion in the middle called visible light. That's what we can actually see with our own eyes. So the range of visible light is 400 to 700 nanometers. That's the length of the wavelengths that we can visibly see. So one of the things we're going to be looking at today in particular are viruses, and viruses are between 10 and 400 nanometers. So what does that tell you about them? It should tell you that we cannot see them with our naked eye. So one thing I hope that you are thinking so far is, why are we talking about viruses? Didn't we mention that they are not considered living? Yes, you're completely right about that. But the reason that we are going to look at them and study them anyway is because historically they were thought of as living. That's one reason. But more importantly is the second reason, because they have a huge impact on living things. So let's talk about why viruses are not considered living. Remember, we said that there was this list of things, criteria that had to be met for things to be considered living. And if we look at them, there are three that viruses do not meet. Okay, first of all is metabolism. They don't take care of their own metabolism. They use the host cells for that. They can't respond to stimuli. And thirdly, they do not reproduce on their own. They have to use the host cells' mechanisms to do that. So for those reasons, viruses are not considered living. But let's get to know these viruses a little bit. First of all, they were discovered by Pasteur in 1884. Pasteur probably sounds familiar because when you have pasteurized milk, it's because he came up with a lovely way to keep that milk healthy for you. But he discovered them while he was studying a case of rabies, and he considered them poisons. He knew they existed, couldn't see them obviously because you can't see them with light, but he just labeled them as poisons. And he actually came up with sort of the first vaccination for rabies when he decided to put some in a little boy. Now, as you can imagine, at the time, there was all kinds of conflict about that. What this horrible doctor was doing, infecting this kid with something that would make him sick. And of course, the way that we know that vaccines work today, it actually saved the boy's life. So that's a little bit about that. Now, in 1892, Ivanowski confirmed their presence by studying diseased tobacco plants. Once he extracted the liquid from a diseased plant, he found that he could basically take all of the substrates out of that liquid, but then when he applied it to healthy plants, he then saw that there was disease present, and so he thought of them as invisible things, these invisible poisons that Pasteur had talked about. Well, in 1950s, along came electron microscopy, and we could see things below the visible light spectrum, things that were much smaller. And then viruses were seen for the first time, and their presence was emphatically confirmed. So even though viruses aren't living things, like we've said, they have a lot of impact on the living world. Instead of being called alive, they're called active or inactive, just depending on the state that they're in. All viruses are obligate intracellular parasites, and that's because of some of those things that we talked about earlier, where they require a host cell for their own metabolism and for their own replication. And also, they can't fossilize, so there's no historic record of them. So that makes it difficult in some ways to look at the phylogeny of it, at least far back in history. Similarly to living organisms, we categorize viruses just, again, to have a context to talk about them in. So they're categorized based on their size and shape and the type of nucleic acid they contain. Viruses can hold up to 3 all the way up to 100 genes. And then one of the main classification categories is the presence or absence of an outer envelope. And very similarly to the way we categorize living organisms, they're classified using order, family, genus, and species, and subspecies. Notice that they don't have kingdoms, and they don't have phyla, and they don't have domains. So many are classified based on glycoprotein spikes that are found on that outer envelope. So those are just some things to know about while we start talking about these viruses. Okay, so let's look at the virus structure in general. We are first going to look at the covering of our virus. So every virus particle has a covering and an inner core. So that's right here. Ooh, I'm a little off today with my circles. Sorry, guys. So to begin with, let's look at the covering. All all virus particles have what we call the capsid. Okay, so here's the capsid. It's a type of covering that goes over the genetic material and that sort of thing. So the inner core is going to have our nucleic acids, our RNAs or DNAs, and the protein units, because it does contain a few proteins as well. On this capsid, we can have various fiber proteins attached, and that's how we can distinguish which virus it is. Now, in addition to our capsid, we also sometimes have an envelope present, so some have them and some don't. But this is the basic structure of viruses. Okay, so here we are with a naked virus versus an enveloped virus. So on the naked virus, of course, we're still going to have our capsid, just like we talked about. That's this shape here. And it covers the nucleic acid on the inside as well as some of the proteins that it carries. In our enveloped virus, of course, there is an outer envelope on the outside. Y'all are going to have to excuse my highlighting. I'm not using a mouse and I'm not great with the touchpad. So if you have questions about what I'm highlighting, I completely understand. Feel free to ask. Okay, so on these envelopes, we have glycoprotein spikes, and those help us to determine what kind of virus it is. So just as an example, the influenza virus has an H glycoprotein and an N glycoprotein, and there's a whole bunch of different combinations that then give you different strands of the influenza. So a while back, lots of people were worried about the H1N1 virus. That's an influenza virus that has a very specific H glycoprotein and N glycoprotein. So here's just a funny story for you, because my husband works in a hospital. They call that the Heine flu, because if you look at it when you write H1N1, Heine flu. Just a little humor for you for today. So viruses, the way that they synthesize proteins, the way they reproduce, is by hijacking the host cell's protein synthesis machinery. And a lot of times this can completely kill the host cell, which leads to all the symptoms that you're familiar with, all the runny nose, the sneezing, the coughing, all those things. So here are just a few viruses that you might be familiar with. Obviously the influenza virus, herpes simplex virus, which causes smallpox, and it also causes multiple sclerosis, which I actually was not familiar with until I started doing some research on this. And it also causes cold sores. There's the human papillomavirus, and there's the varicella zoster, which is another herpes strain, which is responsible for chickenpox and shingles. So viruses are very specific to their host. The specificity is determined by the molecular structure of the capsid or by those glycoprotein spikes that we talked about on the envelopes. The viruses bind to targeted receptors on the membrane of the host cell. So those glycoproteins may have specific cell receptors in your cells. And so when we are trying to stop the viral infection, we use antiviral treatments that normally are aimed at blocking that receptor substrate relationship. So anytime you have an antiviral medication, that is what they are aiming for. Reproduction of a virus has 5 steps to it. They're pretty easy to follow. They should make sense. The very first thing, of course, is attachment, and remember that's specific to a particular receptor site on the host cell. Then there's penetration. The host cell either envelops the virus or the virus just injects its genome into the cytoplasm of the host cell. Either way, it's penetrating the cell membrane and getting into the host cell. The third step is biosynthesis. And that is when new viral components are synthesized using the host cell's ribosomes. And then the fourth is maturation. Once all of those components are made, then they are assembled into a new virus within the host cell. And then step 5 is the release of the new virus as it goes outside of the cell, and it can do that either through lysis, which completely kills the cell, or through exocytosis, which basically the host cell packages it out and sends it out. So bacteriophages, these are viruses that are specific to bacteria. They parasitize the bacteria and infect it and use its machinery to then produce more viruses. And so we're going to look a little bit about the virus life cycle within a bacteria. Okay, bacteriophages have 2 actually different life cycles, but as you'll see, all those components we talked about before are still present. So just like before, we start with attachment, and that, remember, is very specific to the host cell. So here is our attachment process. Okay, and then our next step is going to be penetration. Now, bacteriophages almost always just inject their DNA into the cytosol of the bacteria. So that is the type of penetration that we're talking about here. Now, at this stage, the virus can send the cell in 2 different directions, and that determines which cycle we go through. The first option is immediately after penetration, we go to biosynthesis. This is in the lytic cycle. And that means that the viral components are synthesized immediately after injection. And then we continue through into maturation, where those viral components are then assembled into new viruses, and then we complete it by the release. The new viruses leave the host cell. That's the lytic cycle. Now, The other option is that after penetration, there's what's called integration. And integration is where the viral DNA literally is integrated or becomes a part of the bacterial DNA. And when we're in this process, there can be a waiting period. So once that viral DNA is inserted, that combination of viral DNA and host cell DNA is called a prophage, and that's written right down here, prophage. Okay. Once the prophage is made, then the virus just waits. It waits till that cell is ready to replicate. It waits till those conditions are right, and that can be anywhere from a week to several years. Once the host cell begins to divide, then so does the virus, and so all these daughter cells are made that have multiple copies of the prophage, and so then we can move into the rest of our stages where we have biosynthesis, where that viral DNA is being translated into viral components, and then back through maturation, where the viral components are made into new viruses, and then through the release cycle. So our lytic our lytic cycle is that first yellow cycle, and the red is just an offshoot of that includes an extra couple of steps, and that's called the lysogenic cycle. So if you would go to this other YouTube page and take a look at this video, it really does help explain the lytic cycle and the lysogenic cycle quite nicely. So definitely take a look at that. So this is just some neat information for you. Sometimes the presence of a prophage, remember that's where the virus DNA is inserted in with the host cell DNA, can cause the bacteria that it's in to produce a toxin. So this image on the left is an infection with Streptococcus, which is a bacteria that causes strep throat. Well, if that bacteria has been infected with a virus and it starts producing those toxins, then we get what we refer to as scarlet fever with all these red dots. So what about the reproduction of viruses in animal cells? They reproduce very similarly to bacteriophages. The main difference is that penetration doesn't involve the injection of the viral genetic material. Instead, the virus tricks the animal cell into engulfing it and bringing it in. So the naked virus tricks the receptor into letting them in, and the enveloped viruses can either trick the receptor or they can directly fuse with the membrane, which is endocytosis. Keep in mind that they still have to be very specific to the host. They have to have a receptor. Okay, once the virus is then inside the cell, the animal cell has to uncoat it. The capsid and the envelope are removed. So it's like saying, take your coat off, stay in here a while. And then the DNA or RNA is then free to enter either the lytic cycle or the lysogenic cycle. Just like we saw before, all of that still takes place within the animal cell. Typically speaking, highly destructive viruses enter directly into the lytic cycle. An example of that is Ebola, if you remember when that took place. Other viruses, more like herpes, are more slow-acting viruses, and they enter through the lysogenic cycle, because remember, they can have that long waiting period as well. So if you remember the outbreak of Ebola, because it wasn't that long ago, it utilizes the lytic cycle, not the lysogenic cycle. And it's very destructive. It attacks a lot of the blood clotting cells so that your blood no longer clots, and then that causes excess bleeding, and people can die very rapidly. So I do actually have a good friend who lost his entire family to Ebola and could not go back to his home country because of that situation. So yeah, these viruses and these bacteriophages can be extremely deadly. But this one in particular, not a bacteriophage, it was a virus that directly attacked animal cells. Okay, so the end of either the lytic or the lysogenic cycle results in the viruses being released. So in animal cells, that can take place in one of three different methods: either lysing, exocytosis, or budding. And those are each seen down here at the bottom as you go from left to right. And any of those methods can be used to release the virus from the host cell. So here are just some cartoons to show those three different methods. Lysis usually means disrupting some of the plasma membrane pumps and channels such that the cell fills up with water and literally fills to bursting, bursts open, releasing all of the new viral particles. Exocytosis is sort of a gentler method. The viruses are encased in vesicles that then fuse with the cytoplasm and then are released from there. That's exocytosis. And then there's budding, and this budding, actually the plasma membrane becomes part of the new envelope for the new viruses. So that's the process that is normally used for enveloped viruses. So there's a subcategory of viruses called retroviruses, and they're a type of animal virus, and they contain RNA, not DNA, but that's not what makes them unique. Other viruses do contain RNA instead of DNA, but the way that they replicate using reverse transcriptase and another molecule called integrase is what makes them unique. So if you look over here on our lovely diagram, you'll recognize all the steps as before. We have attachment, so it still has to have a recognizable receptor, so it's a very specific interaction. Then there's, well, they're calling it entry, what we've called penetration. And then we do reverse transcription of the RNA. The viral RNA using the host cell's reverse transcriptase, or sometimes the virus itself has reverse transcriptase that it has brought into the cell as well. That makes a complementary DNA, or what we call cDNA. And then that cDNA enters the nucleus, and we have the proviral portion put into the DNA. There's our prophage, like we've talked about before. And then, just as normal, we carry out the rest of the reproductive cycle of the viruses: the biosynthesis, the maturation, and the release. So the big difference for retroviruses really comes in right here. And retroviruses that you would particularly be familiar with is HIV. So the replication of that viral genome that makes retroviruses so unique follows this order of sequence. The RNA is converted to DNA using reverse transcriptase, and that new DNA again is called complementary DNA, or cDNA. And then that cDNA is used to make a double-stranded DNA that's inserted into the host DNA. Then when the host DNA is replicated, the viral DNA is replicated as well. So here's the thing that makes it a little bit different. When and if the host cell DNA is transcribed, then new viruses are produced. So what that means is that there is a long latency period for a lot of these viruses. So somebody that gets HIV may not show symptoms for a very long time because the host DNA may not be transcribed at that point in time. So that's often why, at least from a human perspective, we want people to be tested because they may not have shown any symptoms yet, but they may be carrying the virus and it just hasn't been displayed just yet. And that means that the the virus can be contracted by other people as well. So if for some reason this video does not automatically load in your browser, take a moment to open a second window and place the address into your second window, because it's really a great description of how HIV infects the cell, how it replicates, and it'll give you a good visual for how that works. So take a minute to look at that video. So I just wanted to mention a little bit about what we call emerging viruses. Those are, every now and then you'll see like this surge in new viruses or viruses that we haven't seen in a long time. And what causes that sudden emergence of viruses? Well, first of all, you can have a genetic mutation, such as in influenza H1N1 or in HIV, that allows it to enter new hosts or become more viable. One thing I didn't say with our HIV that I should have is reverse transcriptase is very bad at correcting anything that went wrong. So it means that it can easily have mutations in the virus. And so actually that's one of the things that caused a lot of problems with trying to treat HIV. Was not only was it a retrovirus, but because of that retrovirus quality of using reverse transcriptase, it was constantly having new strains coming about. So you may have come up with an antiviral medication to one strain, but then it would mutate into something else, and so it's very hard to get under control. Okay, so that's genetic mutation. The second thing is a geographic range is extended. So you have a population that can carry a virus, and maybe their immune system's fairly good at keeping it in check. Well, particularly with us now as a global society, people are constantly moving to new areas and being exposed to different viruses that they may not have been exposed to before. And now all of a sudden you have a virus that is in a body that doesn't have a lot of competition, and so you get that sudden emergence of that virus. So in the next two slides, I just want to touch on viroids and prions because they are mentioned in your textbook. There's still a lot to be learned on these two things. Viroids are the smallest known pathogen at the moment. They were discovered in 1971. They act like a virus, but they don't have that capsid that protects the genetic material. So they are just a simple circular RNA, but that RNA directs its host cell to produce more of these viroids. They don't code for proteins, just more and more viroids. And they cause damage by silencing the host cell's RNA. And this is particularly they cause infections particularly in higher plants. So that, as far as we know, is the smallest known pathogen at the moment. So the other small pathogen is the prions. And unlike the viroids that are just RNA, prions are just proteins. Proteins that are normally present in animal cells, but they take on different configurations. So if you have taken organic chemistry, or maybe you haven't, but you'll get there, proteins have different shapes based on various chemical bondings. And so these prions are sort of like mutated versions of normal proteins within a animal cell. So once the prion infects a tissue, it has like a domino effect, converting the normal proteins into more prions. And these are what we call TSEs, which is a neurodegenerative disease that destroys nerve tissue in the brain. And an example of that is mad cow's disease. So at the moment, we don't have cures for these. Like I said, these are sort of the newest of the pathogens that people are learning about and studying to see what they can do to fix them. But anyway, so today we've been talking about things that are not living things, but they certainly have an effect on everything that's living, everything from viruses, retroviruses, viroids, and prions. All of these affect the living world.